MCAT 2017 Cram Biochemical and Biological Foundations of Living Systems. Passage 4, Typhoid Toxin. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. Typhoid fever is caused by the ingestion of food and water contaminated with the gram-negative bacterium Salmonella enterica cerebar typhi. In 2010, it was estimated that typhoid fever caused approximately 190,000 deaths. Typhi bacteria are deadly because they produce a unique type of AB toxin. Typically, AB toxins consist of two subunits. The A subunit is the active toxin, and the B subunit binds to cell receptors, triggering endocytosis. Typhoid toxin is known as A2B5 because there are two different A subunits, CdtB and PLTA. So here goes the two different A subunits, and a pentameric B subunit, PLTB, I guess, comprised of 5 PLTB peptides. Okay, so this is figure 1 depicting the typhoid toxin A2B5. Figure 1. Typhoid toxin complex shown in a ribbon cartoon structure. The two top proteins are the A subunits, while the bottom ring of peptides is B. So the two A subunits, CdtB and PLTA, while the pentameric subunit on the bottom, PLTB. Okay. Paragraph 2. In epithelial cells, typhoid toxin binds to protocalyxin-like protein 1, PODXL, receptors. Researchers discovered that by binding, introducing shRNA, that's short hairpin RNA, the toxin binding ability of epithelial cells decreased. In addition, they found that when typhoid toxin binds to other cell types, the commonalities are carbohydrate moieties on the surface of glycoproteins. After removing these surfaces from the proteins, the binding ability of typhoid toxin was reduced. Researchers also performed 2 more experiments on typhoid toxin. Experiment 1. Since the A subunit contains 2 different peptides, CdtB and PLTA, researchers attempted to determine which subunit or subunits cause typhoid toxicity. Unlike humans, mice do not have the receptors needed to begin endogenous production of typhoid toxin, but nevertheless respond to the toxin itself. Researchers took mice and injected them with concentrated levels of normal typhoid toxin control, PLTA only rather, CdtB only, or PLTB only, okay? As a control, researchers also observed mice not injected with toxin. To measure the reaction, researchers used weight loss, a symptom of typhoid fever, as a gauge for toxicity levels. Here you can see low weight loss, okay, and high weight loss. All right. Figure 2. Mice were exposed to concentrated serums of various typhoid toxin subunit proteins and to no toxin. The weight loss was recorded in response to these various exposures. Weight loss is a symptom of typhoid fever and is believed to be an indicator of the presence of typhoid fever. Each dot represents weight loss of a mouse. The long horizontal line in each cluster represents the average weight loss, while the smaller lines are the standard deviation from each cluster. Okay? All right. Experiment 2. While investigating the structure of typhoid toxin, researchers discovered a disulfide bond in the junction between PLTA and CdtB, the two A subunits. Once the disulfide bond was broken, typhoid toxin with the missing disulfide bond had a similar cell cycle profile to cells not exposed to typhoid toxin. Okay, so here we see the cell cycle, unexposed, normal typhoid treatment, and treatment with the broken bonds, similar to untreated cells. Okay. Figure 3. The untreated distribution of cells is the usual distribution for healthy cells. Okay. The normal wild type of STV toxin causes cells to undergo cellular arrest, halting the G2 phase of the cell cycle. These arrested cells are unable to divide and grow. The CdtB altered cysteine 269 version of the STV toxin has a missing disulfide bond. And here we see the cell cycle of CdtB altered cysteine 269 version of STV has a similar cell cycle to a normal distribution of healthy cells. Okay. All right. Of the typhoid toxin is most likely the actual cause for typhoid fever. Is it A, CdtB? Is it B, the entire toxin? Is it C, CdtB and PLTA, the entire A subunit? Or is it D, PLTA? I'll give you a moment to think. There were several pieces of evidence that point to the correct answer choice, okay? All right. Okay, so in figure 1, the control cells, this group right here, not exposed to the toxin, have the same weight loss as those mice exposed to PTA only, okay? Matter of fact, PTA only seems to be a little bit healthier than no toxin at all, okay? Mice exposed to CdtB, this portion of the A subunit. Okay, so mice exposed to the CdtB toxin have the same weight loss as mice exposed to both. Okay, to the full typhoid toxin. When I say both, I mean both the A subunit consisting of the two subunits and the B pentameric subunit as well. Okay, so full toxin represents this right here. CdtB only, the blue portion. Okay. All right, so now that we've established that, since CdtB produces almost the same result, okay, by itself as does when combined with the other proteins,
See, when we have CdTB and PTA, the full A subunit, there's a significant weight loss above normal as well. And here we have the full toxin. CdtB is therefore most likely the candidate for the toxin responsible for the symptoms involved in typhoid fever. Okay, so the correct answer is answer choice A. All right.
What is the most likely explanation for how short hairpin RNAs, shRNA, reduce the toxicity of typhoid toxin? Is it A, the shRNA molecules are inhibitors of PODXL gene transcription? Is it B, the shRNA molecules bind with the A subunit of the typhoid toxin? Is it C, the shRNA molecules bind with the mRNA for PODXL receptors? Or is it D, the shRNA molecules block PODXL receptors on the cell surface? I'll give you a moment to think. You may want to refer back to the passage where the shRNAs or short hairpin RNAs are mentioned, all right? Okay, so let's break down the reasoning behind the correct answer choice. shRNA and RNA strands are produced intracellularly, meaning inside the cell. So after a shRNA molecule is produced, the loop of the hairpin is basically cleaved and two short strands of RNA can separate, okay? Once separated, the two short strands of RNA can hybridize to other strands of nucleic acid sequences that are floating around in the cytosol of the cell. The readily available strands of nucleic acid inside the cell are mRNAs, messenger RNAs. mRNA is abundant and also single-stranded, so it will bind quickly to the short single strands of shRNA peptide chain. Okay, and hence it's blocked. So it's most likely that the short hairpin RNAs contains a complementary sequence for the PODXL gene, okay, and therefore blocks PODXL receptors from ever being made in the first place in the or on the surface of the rough ER, the rough endoplasmic reticulum. So without PODXL receptors, the cells will not bind the typhoid toxin. So basically, the shRNA molecules are inhibitors for PODXL at the level of gene transcription. As the Bible says, they dried it up from the root. So it never came into existence. Okay? All right. Where is the most likely place where surface glycoproteins are glycolated? Is it A, in the endoplasmic reticulum, B, in lysosomes, C, on the cell surface of the plasma, basically the plasma membrane, or D, in endosomes? I'll give you a moment to think. This requires standalone outside knowledge apart from the passage. All right. All right, so hopefully you've arrived at the correct answer choice. If not, that's totally fine. Let's get into it right now. So glycoproteins are mostly located on the outer plasma membrane surface, okay? This isn't necessarily a glycoprotein depiction, but this is the outer surface of the plasma membrane. So if you were to zoom in, you would see glycoproteins, okay? Just imagine. So for a protein to be located on the cell surface, it had to undergo exocytosis via the snipping off of a vesicle, okay, as you can see here. Vesicles for surface proteins are formed in the rough endoplasmic reticulum. This is where they begin their journey. So all modifications to protein will typically begin in the rough endoplasmic reticulum and continue in the Golgi body, okay? Modifications are not made in the vesicle during exocytosis, so the correct answer is the rough endoplasmic reticulum. So in the rough endoplasmic reticulum, these glycoproteins are glycolylated, or in other words, a sugar group is added, okay? All right. How does the breaking of a disulfide bond change the typhoid toxin? So here goes the disulfide bond that would be broken between two cysteine residues, okay? Would it be A broken disulfide bond alters the tertiary structure of the protein and inhibits normal binding to cell surface receptors, thus eliminating the toxicity? Is it B, the breaking of the disulfide bond decouples the toxic protein from the typhoid protein complex, yet the toxic protein itself retains its toxicity and causes cellular arrest? Or is it C, the newly created terminal sulfides have different balances for binding and although will not bind exactly to cell surface receptors, they will remain mildly toxic to some cells. This is like a whole nother passage. And last but not least, D, the disulfide bond is the main active site of the protein, so the protein is unable to make toxins that will bind to cell surface receptors. Therefore, there is no toxicity. I'll definitely give you a lengthy moment to think. This requires some outside knowledge apart from the passage. Although you could use, well, you would have to infer and really be in tune with the passage. You could potentially answer it from the passage alone. All right, so hopefully by now you were able to press pause and arrive at the correct answer, but if not, that's completely fine. Let's take a look at the solution, or the mental thought steps into arriving at the correct answer choice rather, because this isn't like a technical mathematical problem. Okay, so basically, no lengthy. I get so flabbergasted with that word, if that's even correct. Okay, so because basically the correct answer choice is answer choice A. So a broken disulfide bond basically alters this tertiary structure that you see here. Now this was mentioned in the review list for the MCAT as prescribed by the AAMC. You had to know, the importance of tertiary, quaternary, primary, secondary structure. So it alters or disrupts the tertiary structure of the protein and inhibits normal binding to cell surface receptors, thus eliminating the toxicity. Okay, this is pretty self-explanatory. Everything else is just way too wordy and way too incorrect, right? All right. To further validate the findings of experiment 1 and make more direct connections to the physiology of humans, researchers should perform which one of the following experiments? Is it A, use immunofluorescence staining to observe typhoid toxins intercellular interactions in mice and observe locations of binding? Is it B, design a mouse model that has receptors for Salmonella typhi uptake along with engineered Salmonella to secrete modified toxins? Or is it C, inject mice with concentrated toxin and observe additional symptoms of typhoid fever beyond just weight loss. All right, this is a tough one. I'll give you a moment to think. All right, let's not waste any time. I'm sure your test is coming up soon. Even if it's 90 days away, that's way too soon. Okay. Okay, so the minor problem with experiment 1, if you go back and refer to the passage, is that an observed correlation with weight loss doesn't necessarily mean, the host is infected with typhoid fever. It could be due to some other cause, okay? So weight loss and typhoid fever in the mouse or mice, these two things are not mutually exclusive. However, moreover, mice don't produce typhoid toxin endogenously, like they can't do it on their own. Like once infected, they can't perpetuate the toxin. So ideally, to compare the physiology of mice to that of humans, scientists should use a model that produces typhoid toxin endogenously, okay? So they would have to come up with some genetically altered typhoid toxin that could do this. So the best solution basically is to study typhoid toxicity in humans to use an analogous mouse model. And in actuality, in 2012, a mouse model was developed that has human-like receptors so that mice would produce the toxin endogenously, okay? So the correct answer choice is answer choice B.